So today I'm going to be talking to you about the question I'm addressing for my dissertation, which is essentially, can character displacement drive the evolution of root traits? Before I dive into this specific question, however, I want to provide some context and talk a little bit about a pervasive challenge in the field of evolution and ecology, which is understanding the relative role of competition in the evolution and distribution of closely related plant species. So why is this such a pervasive question? Well, theoretically, no two species can compete for the same resources and coexist. Well, closely related species, because they have shared evolutionary history, tend to be very similar in resource-associated traits, therefore overlap more in their niche space, compete more intensely. So based on this, it's been often predicted that closely related species should not co-occur, because it would eventually lead to the competitive exclusion of the weaker competitor. Despite this, however, we see that natural populations of closely related plants not only commonly co-occur, but that they tend to be more phenotypically divergent in regions where they're found together relative to regions where they're found apart. So one possible explanation for this observation is the process of character displacement. So this is a process essentially in which two species evolve in their resource-associated traits as a response to selection from competition. So initially what happens is two species come into contact, they have overlapping distribution in a resource-associated trait. However, selection favors those individuals in the extremes. And so what's thought to happen is that after various generations, selection favors these extreme phenotypes. The means of this resource-associated traits is diverged, and these species no longer overlap so greatly in niche space. And so this has been thought to be very important in enabling species coexistence, but also leading to the evolution of novel traits, and can potentially also lead to adaptive radiation and speciation, as been the example of the Galapagos finches here. So since it was first introduced in the 1960s, evidence indicating the importance of this process has increased, but most evidence lies in animal systems, whereas is this an important process in plants remains to be further studied. If we zoom in on the traits that have been studied in plant systems, we see that most traits looked at have been above ground and looking at above ground interactions. For example, competition for pollinators and acting on floral morphology and color. Yet despite its importance in uptake of resources from the soil and mediating below ground competition, to my knowledge, no study has examined how this process may be influencing the evolution of root traits and therefore explaining patterns of plant diversity. So my goal for my dissertation is to kind of address this gap by looking at whether or not this process is viable. Can it lead to the evolution of root traits? But before I go into talking about that, I kind of want to move into why you should be looking at root traits. Why are they important? Well, first of all, as I mentioned, their primary function is the uptake of resources from the soil. But they also mediate below ground competition, and that could be detrimental to plant fitness and ability to establish successfully within a plant community. Further, they're super important to the carbon pool and cycle. So as you can see, for many plant species, most of their photosynthate is actually allocated in the below ground biomass. Furthermore, processes such as root respiration and exudation contribute to the terrestrial biosphere's largest carbon dioxide efflux into the atmosphere. They also host a myriad of beneficial microbes, and this is incredibly important to soil fertility. For example, many leguminous species form mutualistic interactions with rhizobia, and this is important for nitrogen fixation. Lastly, many roots are actually important to our crops because many are tubers, such as sweet potato, which we depend on. So it's not only likely that roots have responded to selection from competition, but understanding how they may have adapted can have important consequences in understanding plant community patterns, ecosystem functioning, and lastly can have some kind of application to crop improvement. So the way I'm characterizing root traits is essentially through morphology or architecture. Morphology is more talking about measurements done to individual root traits, such as root length, root surface area, etc. And architecture just takes into account all these morphological traits in space, which is incredibly important because the soil is a heterogeneous environment. So depending on architecture will determine whether or not a plant has accessibility to a particular nutrient. And so, again, I want to reiterate, I'm interested in testing the process happening in root traits. And so, to validate process, I want to use this metaphor that the process is a driving car in which if the criteria are validated, it will run. It will influence the root traits. So the first thing I have to demonstrate is that the phenotypic variation under selection has a genetic basis, right? Because then it could be passed on throughout generations. And that is analogous to having fuel for this driving car. Secondly, I have to demonstrate that when we have two plants competing, the extreme phenotypes are favored. And that is analogous to having a functioning motor in this metaphor. And so as a preliminary experiment, I looked at whether or not we have this fuel for the process by addressing these two questions. How do closely related plant species vary in root traits, and is there evidence for genetic basis in those traits? I'm using a morning glory study system, Ipomea heteracea and Ipomea purpurea, because first they're sister species, secondly, they're fast growing, thirdly, they're annual, so I can have fitness estimates within one growing season, and lastly, they're diploid, so that eases any genetic analysis I want to do in the future. They're globally distributed, and they commonly co-occur in the eastern United States. I'm working with 4 populations in Pennsylvania where they were found occurring together and alone. And this is just a preliminary study I did from populations in the US of these two species, where I did a PCA of environmental variables, including temperature, precipitation, soil, and climate. As you can see by the overlap in this environment space, they would suggest that they have similar habitat use. So they're a good model system. So, as I mentioned in my preliminary step, I wanted to look at root trait variation and is there evidence for genetic basis. So I did a common garden where I grew these plants in rhizotrons. What's a rhizotron? So a rhizotron is, this is a custom made box, thin box made of plastic with a transparent sheet in the front that allow you to place the plants in soil so it resembles their natural environment, but also without destroying the plant. You can watch the roots grow in situ. And so this is what it looked like set up in the greenhouse, and after a couple of weeks, this is what the root system looks like, and you can kind of appreciate its architecture and morphological traits. And so in a nutshell, I looked at 6 maternal lines for both species and across 4 populations and replicated this 3 times, and then I measured the root traits about after 2 weeks of growing them in image J. And so what are my results? For the first question, how did they vary? Oh, sorry. So before I go into that, the traits I looked at was root angle. So root angle is essentially the angle formed between the top most lateral root and the primary root. It's a good indicator of architecture. I looked at lateral root number, I looked at primary root length, root system width, and total root surface area. Here we are. And so how did these 2 species vary in these traits? Well, I found considerable and significant variation between species. And despite that most traits are significantly different between species, as you can see by the overlap in box plots, that there's still conserved variation between them. I further did a PCA to kind of take into account all these traits simultaneously, and I found that in the first dimension, indicated by the bar graphs on the right, the 2 traits that loaded most strongly were primary root length and root angle, and that they're inversely related. So what this indicates to me is that as we move to one direction, we have root systems with shorter primary root lengths and more obtuse angles, whereas as we move on the other direction, we have the opposite. And so this is a good indicator for root architecture. In the second dimension, I saw that primary root length, total root surface area, and root system width loaded most strongly, and this is a good indicator for root system wise. So as demonstrated by the overlapping ellipses of these 2 species, they have a lot of variation in root architecture and root size and overlap. And so for the second question, what root traits varied by maternal line, I found that root angle, primary root length, and total root surface area had either significant or marginally significant maternal line effects. And this is incredibly important because it indicates that these traits might potentially respond to selection. And so my conclusions from this preliminary study is that first, these root traits vary considerably between these 2 species. There is overlap and there are species differences. And secondly, and probably most importantly, is that there is some evidence for maternal line variation. So we kind of suggest that we have fuel for the process of character displacement. So the second step is to see, do we have the motor? So is there evidence that selection is favoring more divergent phenotypes? How is variation in root traits between competitors influencing fitness. So I'm addressing the question, does similarity impact fitness? And to do that, I've been doing a large field study. For those of you at MFI might have seen me running around this large plot there. So it just finished, but the purpose of the study was essentially to look at these plants competing and alone. And so what I did was I drew 8 maternal lines per species within just a single population, alone or in competition, for all possible maternal line by species combinations, pairwise combinations possible. And so basically, the first part of the study is to see how these root traits vary when they're grown in the field. And so I dug up half of the plants I had out there to phenotype the root systems. You clean it up, you get kind of a soil core of them, and then you photograph like parts of the root system that you've dug up, and then I characterize them similarly as I did in the greenhouse study. And then the other half is to look at fitness, and essentially I left the other half of the plants to measure relative growth rate, flowering time, flower number, and seed set. And so the ultimate prediction is, as if this process is in fact important, I would expect to see that individuals competing with more divergent phenotypes will have higher fitness relative to individuals that are more similar to one another. And so you would expect a positive trend between a dissimilarity index and fitness. And so, as my takeaway message, by learning the adaptive value in which root traits may have evolved, particularly competition, can teach us about the context, right? And so this can funnel into and provide us more information to answer this even bigger challenge and pervasive question in the field of how competition between closely related plants may influence their evolution and distributions. And with that, I would like to acknowledge the members of my lab, the staff at MFI, volunteers, and my funding sources. You can follow me on Twitter if you're interested in seeing updates on the project. Thank you for your attention, and I'll take any questions.
Questions for Sara?
Thank you. That was a great talk. I know you're not working with perennials, but I wonder if you might speculate, because it's interesting you're doing all of this work with short-lived annuals, and I'm wondering if you think that phenotypes would be expected to be even more divergent where you have competition that lasts for longer periods of the year or multiple years, and more extensive spatial areas.
Interesting. So definitely depending whether you're a perennial species or an species, there's already differences just between those two categories in root traits. These species are more short-lived, like you would see more fine roots, etc. And that impacts how they take resources up. But in terms of whether or not you would expect to necessarily find more divergent patterns is a tricky question too, because then you have more generations occurring with annuals, right? Faster generations. So there you have that factor that might impact how quickly or how intensely divergence might happen. So I'm not really sure if I would necessarily expect species that are longer lived to see more divergent patterns. If anything, I would expect the time scales to just be different. Does that make sense?
More speculation. In traditional agricultural systems, people often intercrop. They put within the small land area, they plant different species or different crops very close to each other. Have you thought about or looked at the evolution of traits within agricultural systems?
So I have thought about it, and I have looked at some literature, and I've read that there's this hypothesis that differences in root architecture, for example, between soybean and maize, and I believe pumpkin or a species family of pumpkin is something that's been intercropped for many generations in Central and South America. Mm-hmm. And there's a hypothesis that potentially this is why you see increasing yield when you're intercropping versus having a monoculture. That is these differences in architecture. And I have read a study that they ran, it was a simulatory study where they were able to take parameters of the different architectures of these plants and compared monocultures versus these intercroppings. And there is evidence, at least at that theoretical level that differences in architecture between these species is, and what's the word, leading to the increase in yield. Yeah.
Thanks.
Hello, could you please explain a little bit more why the same environment would simultaneously favor two completely different extreme traits? And after the character displacement is established, do they still compete for the same resources, or did they differ for the resource competition as well?
Okay, so two questions. First question, correct me if I'm incorrect, why would I expect the extreme phenotypes to be favored within the same environment of these two competitors? So that's just based on theory. We're assuming that these fitness differences between these individuals are the same when they're in this environment, and that the only difference is that if they're growing with their competitor, competing for that same, because we're assuming, right, that they're competing for the same resources, that would be the big factor impacting their fitness. So you'd expect that if these individuals, if there is this correspondence between traits and root traits and the resources they're obtaining, you would expect that individuals that are very similar to have lower fitness relative to individuals competing with less similarity, because there is that form and function relationship. So you would expect extreme phenotypes to be favored because those individuals more within the overlapping region are the ones that are maybe in a statistical standpoint suffering more relative to extreme phenotypes. Does that make sense? And for the second question, I'm sorry, I think I forgot. May you repeat it?
Thanks. So after the character displacement is established, do they still compete for the same resources? So is the problem solved or not?
Oh, well, that's what we're kind of testing. This is trying to set up the experiments to at least suggest that individuals, if there's a relationship between a particular phenotype, similarity, and they are in the same environment, presumably the resources are the same across the different samples that I have, it would at least give evidence that there may be a causal link, that these individuals that are more dissimilar are in fact using different resources. So I guess another way to get more directly to that question would be to alter resources availability, and then do kind of a factor analysis where you look at individuals of different phenotypes and grow them together. And that would allow you to more, I guess with more confidence, ascertain whether or not these individuals are shifting resource use.
All right, thank you, Sara.
Thank you.